Well, hey there, folks. Mr. Bleeker again. So, in topic 24, chapter 25, which is all about DNA and the molecular basis of inheritance, we've covered a fair bit of territory. Lesson 1, we covered DNA history and its fundamental structure. Lesson 2, we looked at the semi conservative replication of this double stranded molecule. And now we plunge into lesson 3, which is transcription of DNA into mRNA. Now, that might sound kind of fancy, but what that really means is looking at the genetic code and making copies of the genes so that we can make all the proteins in our body. But the first thing you have to consider about DNA is where it lives. It lives in the nucleus. Now, I'm just going to draw a little analogy for you here. When you think about DNA, if you haven't seen this before, this is the US Constitution. And it's under glass, and kind of like the Mona Lisa, this is the original, but you can't take the original out of the display case. You can't take the original out of the library, so to speak. But what you can do are take copies of sections, bits of information. And it's not much different from how DNA works. You can make a chemical copy, but the original needs to stay put, and it needs to stay put in the nucleus. So I'm just going to dismiss that there. There we go. So click on transcription of DNA into mRNA. Emphasis on the scribe part. When you hear the word transcription, you hear scribe. And when you hear scribe, you think of somebody who writes or making a message. That's a very good thing. That's what I want you to be thinking about with this lesson. All the instructions, everything's laid out here that you would need. You've got your slideshow here. Nice information all laid out for you here, videos, animations, etc. Several of which we'll look at as we do this sort of lesson and we lay out what transcription is. And go from items 1 through 6. Of course, make sure you do your book reading. If you've forgotten your book, remember in the course at the top that there is a digital textbook format sitting there. So that's quite good. That's searchable. But I digress. We'll get going. Okay. So as an animation, just sort of as a predicate to what we're going to do, what I want to look at is what's going on in the cell. Now that we've done our cellular sort of background, we have a better idea of the parts of the cell. This is kind of neat. I'll just do a slider here. Now, when we go into the nucleus, in the nucleus you have chromatin. Chromatin itself, the little threads of DNA, can be organized into, well, in humans in specific, 23 pairs of chromosomes. And we talked about how the DNA was looped over little sort of protein balls that we call histones. As we zoom in a little bit more, that's what I wanted right there, we can look at the specific DNA itself, the genetic code. Now there's many regions of DNA, and they're what we refer to as coding regions. And if you think about genes, there's genes for eye color. Height actually requires the interaction of many genes to come together and give you your height. Some things can be as simple as one gene, whether or not you have a straight thumb or a bent thumb, whether or not you have a widow's peak, where your hair points down in the front, or whether you have a straight hairline. Some are kind of simple genes, but thinking about the construction of an organism, and if this is the blueprint, there could be many genes at all times acting together. I have green eyes. That's polyallelic. It takes many genes to make that eye color. And it's why my green eyes aren't like your green eyes. They're not like my mom's green eyes. There's just too much genetic copying going on to make these custom proteins in my eyes. So be proud of your eye color. Now, just in a quick sense, way of looking at this, we have little sort of chemical scribes that come along. And in the same sense as copying DNA, one of the things we had to do was unwind it. And once the DNA is unwound, then what we're able to do is get in there, and I really like this slide, we're able to sort of pry it apart. And if you look right here, and I'll just press play for a second. There we go. What you see is the message right there. I'll just back that up a skosh. Right there. This message right here is messenger RNA. Now it's not double-stranded like our DNA molecule here. It's just a copy of one side of the DNA. In other words, we're just reading one side of the track. But that's sufficient to make messenger RNA. Now, in overview, the messenger RNA, and we'll just kind of watch this come together, comes together in a very interesting way. And one thing that you'll notice, and I'll just hit pause. Oh, we lost it. Let's see if I can just pause it right there. As the messenger RNA is being transcribed along this side, you notice the DNA strand right here, and you'll see its genetic sequence. Now keep your eye on that, because normally we think of adenine bonding with thymine, right? These are normally bound together. Cytosine bonding with guanine, right? Those A's with T's, G's with C's. But one little rule that you have to remember with mRNA is that it contains uracil, this right here. It does not contain thymine. That's a relatively new nucleotide, sort of an older one that you see. The old thymine, if you will, humor me here, is uracil. So you have to keep this in mind, because as the codes go on the mRNA, notice I said codes go on, codons, we'll come back to that later, you'll notice that there will be no T's in the sequence. So I'll just press play and we can watch that formulate along there. And there's the single-stranded molecule being formed as we're reading along a specific gene. And there you go. There's a little bit more here that we'll get to. What I will predicate this by saying is that not all of the mRNA is going to be useful. There's some little chunks that we'll have to chop out. These little in-between pieces called introns are sort of thought to be filler, and they get excised, they get removed. It's these exons which are the the important parts of the genetic message. So we'll just keep it simple for now. Okay, I'm going to pop out to our notes and fill in some information for you. And there we go. One moment there. I just have to, apparently I put a code on this. Should be good. Okay. My apologies for the delay. Sometimes I forget even my own codes. If you remember this guy, this is Troy McClure. He was a Simpsons character, sort of a comedy element. It's a little dated. Humor me. I really like this slide because it shows off what I think are the most important aspects of what we're looking at, which is that we look at DNA. Grab a nice blue here. That we look at DNA and we do the scribing part. Now, specifically transcribing, and we write an RNA message. But because it's a message on how to make proteins, we refer to it as messenger RNA. If you think of this whole thing as an act, we know that the ribosomes are our protein makers in the cell. So therefore, the message must insert into our friend the ribosome. And this has always kind of reminded me of, if you're ever little kids and you heard of the Beyblades or those you would put the little key in the spinning top and give it a pull, right? And you'd see a top spin off. Kind of reminds me of that. But this message right here is essentially a template for the correct order of amino acids. If you remember, amino acids are little Lego chunks of protein. So when we eat meals and we disassemble protein into all the different amino acids, when you're lifting weights in the gym or when you were forming in the womb, when it came to make proteins, eye color, hair, skin, bone, muscle, those amino acids had to be put into place in a specific way. So the raw material, the little Lego bits, are the amino acids, and will follow the messenger RNA sequences bit by bit, DNA gene by DNA gene, until we grow up a critter. Now, transcription is the scribing part, but when things actually happen, when the blueprint is finally expressed and we have something real, when we have a protein down here, there we go, then what we can say is we can say at that point that we have translation right here. We have translated a gene into an actual protein. So transcription, translation, results. Sort of the overview of the whole act. Neat little slide. I've always liked this one. Each one of these here is meant to represent different amino acids as they're strung out and lined up to give us a protein. And if I remember my biochemistry correctly, and I do, that protein starts out as sort of long strings in its primary form, and as it's bent and globbed and many more proteins are sort of put together, we start to get the more complex things like hemoglobin, insulin, which would be fourth degree or quaternary proteins. That's really when you've got everything put together and you've made a fairly impressive, large three-dimensional structure. It comes right down to this, folks. The DNA molecule itself, just like when you did semi-conservative replication of DNA, has to be unwound, and the hydrogen bonds have to be basically have to be pulled apart. Hydrogen bonds, as you recall, are kind of weak, static, almost like static bonds. Once we move the two sides of the DNA apart, we have the left and right facing strands, if you call it the 3' to 5' and the 5 to 3, and we can copy, we can make an mRNA message against one side of one strand. So it's all about making a copy of the DNA sequence, and we'll call that mRNA, or messenger RNA, right here. And the name of the game in this case is that message, or mRNA, that copy leaves the nucleus. And I can't stress that enough. That's a very important point. You don't make things in the nucleus. It's very small. Okay, I'm lying, partially. Ribosomes get made by the nucleolus. I get that. But the thing you have to remember about ribosomes is they're very, very small. It's not like there isn't enough room for that. But when it comes to putting together primary, secondary, tertiary, and quaternary proteins, and you start to make things pretty big, that really needs to happen, let me think, out in the cytoplasm, or, oh yeah, in the rough endoplasmic reticulum. See, it all fits together. When you look at DNA and you say, well, what's the enzyme? What's doing the transcribing? Well, then you just named it. You can call the enzyme that does the process of transcription transcriptase. As I recall, ASE is a dead ringer for something well, or an indicator for something that is an enzyme. So this is the enzyme that does the transcribing part. Looks a little bit, I think if you look at it for a moment, you'll think, oh, it sort of looks like DNA polymerase. Remember, we were looking at a couple different DNA polymerases when we were examining the semi-conservative replication of DNA. This is a little bit different. This makes RNA, and they're trying to indicate that to you with this red RNA strand here, which will be released from the enzyme as it moves along the different gene sequences. So you can unwind DNA. All these mechanisms exist, but now instead of semi-conservatively replicating it, now we're making a single-stranded mRNA template. So let's put that in, an mRNA template. That's an important point. When it comes to making a template, if you have your DNA strand, and there are two sides of that DNA strand, one of which will be red, so we'll say that this DNA strand right here is being read. There's no surprises in how this is read, save that, of course, C's go with G's, but A's, instead of going with T's, go with our little friend here, uracil. Now, some students get this backwards. They seem to think that the DNA strand, which is this guy, can't have thymine. Well, it's perfectly acceptable for DNA to have that. So, of course, that matches with adenine. Nobody said mRNA couldn't have adenine. The only thing we said with mRNA is that there was, let's go down here, there is no thymine. So I'll just draw a little Ghostbuster symbol through that. No thymine. Okay, so keep that in mind. So as soon as an adenine here is to be paired, well, what corresponds to that is uracil. Important point. Give you a chance to absorb all that wonderfulness. Uracil is a nucleotide. Just realize that it's an alternative nucleotide that you find in RNA. I really like this picture. This is a little sort of snapshot from your booklet. Now you sort of see, now you realize why the nucleus has pores. Because what you're watching come out of it is an mRNA, and that's always single-stranded. If you're remembering Biology 11, I talked a lot about how RNA could be the code of some viruses, like cold viruses, rhinoviruses, like influenza. It has a single-stranded genetic template. RNA is older. It's been used for a very long time. We've adapted it as a very efficient way to express our genes. So here it is. You can see a single-stranded copy of this popping out of the nucleus. If you examine this, sort of neat little, I have to wait for it to catch up. If you examine this, there's the nuclear pore, and you can see it forming and how it sort of spools. It will bend over on itself, just sort of forming these little loop-like structures. But nonetheless, it's still a single-stranded copy of sort of a gene sequence. This is it. A lot of people will get confused and they'll think, well, geez, like it hasn't gotten to the ribosome yet. Nothing's being made. Well, this is the end of the scribing, folks. From this point on, if you fit into a ribosome and that genetic sequence, which has been copied onto the mRNA, if it's turned into an actual protein, well, that's translation. So this is it. This is the end of transcription. The next step, of course, would be translating that into a protein, specifically a primary protein, one amino acid at a time. Now, you've asked me to keep these brief, so I'll listen to you. Don't say I never do. I'm just going to turn off mirroring. There we go. I have some, as per usual, some nice little animations for you. This one, what I wanted to show you was what's happening. You've got your nucleus of your eukaryotic cell, and the idea that what we're going to look at in the DNA is coding regions. Zoom in on this a little bit. Let's see if I can give you a bit of a better view. So the genes are, of course, here, but you have to understand there's spacers and fillers. Just like when you read words in a book, there's spaces between words and there's sometimes punctuation. Not all of that is the meaning. So there's coding regions here, and then those are called exons, and then you've got these little spacers that generally get cut out called introns. Initially, your mRNA will look like this, and you'll have the green exons and all those little intron spacers that more or less have to go. When the mRNA is tidied up, cleaned up a little bit more, those little white introns are removed from the coding sequence so that you just have more or less the pure gene, exactly what you want to say to the ribosomes. In other words, hey, make a protein like this. It needs to be Bleeker's green eye color, so get at it. It's a lot to do. Nice snapshot here of DNA versus RNA. Of course, we have our phosphate group, and DNA has its deoxyribose sugar. If we go back when we were looking at nucleic acids, right here, remember, the carbon starts here. 1, 2, 3, 4, 5. Okay, or yeah, 5 prime right there. And there is an oxygen missing off of carbon 2 here, whereas in RNA, it's fully oxygenated. That's why this is deoxyribonucleic acid, and this is, well, you'd never hear it said that way, oxyribonucleic acid. No, that's where the D in DNA comes from. Missing an oxygen right there. So the nucleotides, there's 2 chemical families, purines and pyrimidines. So the purines were the AGs, and the pyrimidines in DNA are the CTs. Well, guess what? In RNA, there's no thymine, so there's uracil. Voila, now you know. Uracil is a pyrimidine. DNA itself is a double-stranded molecule. Okay, so as you recall, you will see, there we go, zoom in. You will see triple hydrogen bonds between your cytosines and your guanines. Okay, and you will see double bonds between your adenines and your thymines. Notice the direction of the strands because they run anti-parallel. When you run from the 5 to 3 prime end, okay, that's one direction, and when you go the opposite, you'll notice that, of course, you're going in the opposite direction, so these are upside down. It's still the regular edges with the sugar and the phosphate going back and forth like that with the genetic material in the front. Just a little review. Over here, when we look at an RNA strand, realize that from 5 to 3 prime, it's just single-stranded. We don't do this. There's no double-stranded RNA. So here you go. Polynucleotide, that's kind of fancy talk for a DNA molecule on the left or an RNA molecule on the right. Voila. This is a very good snapshot from Wikipedia. Little geekdom here. SVG stands for put it this way, a file that doesn't blur as you zoom in. This is a simple vector graphic. So on the left, nice visualization, uracil in the orange, and on the right, thymine, instead of orange, they chose a red, single-stranded, double-stranded molecule. Looks good. One more visualization of that. Apparently I wanted to make a point. And here's a neat view of the region called the promoter. See if I can. I'm going to go back to kind of the beginning. There we go. Sorry, I had that queued up. But if there's a gene region of interest, there will be a promoter region that will convince that area to be red. If you take biology, you'll do a lot more of this. Upstream or downstream from the promoter, there's a promoter there, that's where you want the gene to start to be red, and after it is the coding region. And then once you finish the genetic region that you wanted to copy, because that contains all the information, there will be a termination chunk. So RNA polymerase, the enzyme that makes mRNA, now specifically the RNA polymerase is transcriptase. So they're not calling it that, but that's what it is. So in your promoter region, they tend to be TATA boxes or TA regions. So the RNA polymerase will see that. That's a dead ringer for a start of a gene. And it will bind on, and we'll assume that we've got some unwinding already happened by the unwinding enzymes. It will, once it gets past the sort of promoter region, it sort of begin here. And then this is where it goes to town with the mRNA sequencing. I mean, you can see the gene copy being read and being transcribed. There's a very, very good visualization of this. I like it quite a bit. Until you finally hit that terminator. That terminator region just is sort of like the transcriptase enzyme will just get knocked off. Bam, there it goes. And that is your mRNA sequence, and the end sort of flops over on itself a little bit here. That tends to happen with the terminator sequences. So it's quite good. Another view of transcription. This is pretty good. This is on the course site as well. There you can see those that TATA region there. You notice the nucleotides, and there you go. Until a terminator is hit, and then off pops DNA polymerase. Please don't think that this is enough genetic material to make some elaborate protein like insulin. There are lots of genes that have to be read. It's not quite as simple as just read one section. I think this is just overkill. This will come up a little bit later. Okay, so I'm going to call it, that's about as short as I can do this, folks, for transcription. Thanks for listening. Next stop, translation. That'll be out at the ribosomes, either out in the cytoplasm, or we'll be heading on over to the rough endoplasmic reticulum to get the job done. Thanks, folks. Have a good one.